Hello, hi, a very happy good evening. So this is Krishna Ma'am. We are talking about the story of life on earth, and that's biology. So welcome to my new beginning series. Today we're going to talk about the next part of your sexual reproduction in flowering plants, that is Megasporangium. So last week, that is day before yesterday, on Monday, we spoke about Microsporangium. So today we're going to talk about Megasporangium. So what is this Megasporangium all about? So is it a very huge topic? Is it a very tough topic? So definitely it is no to both. So it is not a very huge topic, or it is neither a very tough topic. It's a very simple one. Hardly in the next 15-20 minutes we'll be done with this topic. So this sexual reproduction in flowering plants talks about how plants go forward with their sexual reproduction. So for plants to have sexual reproduction, they need pollination, right? So even before pollination, they need to produce the gametes, the male gamete and the female gamete. So who produces the male gamete? The andrecium portion, so especially the stamen. Now we spoke about how the andrecium produces the male gamete, the pollen grain, and how the male gamete is released, right? So today we are going to see how the female gamete is designed and how the female gamete is going to produce the female gametophyte. So that is all the topic for today. Hi Natasha, hello, a very happy good evening. So thank you so much for joining my lecture today. So I hope you get benefited from this lecture. So thank you once again for joining us, and please don't forget to like, share and subscribe. Fine, so let's move forward. So we're going to talk about Megasporangium. So before we move forward and talk about Megasporangium, so very quickly in the next 5 minutes, I will give you the recap about Microsporangium so that you have an understanding of what happened in the previous lecture, right? So here we move. So we're going to talk about the male portion. So the male portion is your andrecium. So the andrecium is represented by stamens. So the stamens have two parts. One is filament and the other one is anther. Right? So the structure is somewhat like this. Right? So this is your filament and this is your anther. So the proximal portion of your filament is the receptacle or the thalamus. So this is the receptacle. So receptacle is the place from where all the four floral whorls of a flower arises. So are you able to recollect it? Yes? Okay, so Abu Deepa, hello, a very happy good evening. So I guess you're new to my lecture, so thank you so much for joining my lecture. So please do like, share and subscribe. So this is the receptacle, and towards the distal portion, so we have the anther. So the size or the length of the stamens, the filaments keep varying. It is not fixed in all the flowers. Now talking about the structure of your anther. So as you can see, anther has two lobes, right? So this is one lobe. So this is the other lobe. It has two lobes. So inside these two lobes, there is a thecal layer. So this thecal layer is the wall layer. Right? So a thecal layer separates each of the two lobes further. Now if I cut open the anther like this, so I have a structure like this. Right? So I have four corners. So in each of the four corners, I have what is called as microsporangium. So this is known as your microsporangium. Sorry, this is known as your microsporangia. So this is known as your microsporangia. Your microsporangia is also known as the pollen sac. Fine. So what is the function of the pollen sac? To produce pollen grains. So since there are four microsporangia, so it is known as tetrasporangiate. So this is known as tetrasporangiate. So this microsporangia is surrounded by walls. So the wall of this microsporangia is the microsporangium. Fine. So this microsporangium wall has four layers. So the center portion is microsporangia. It is surrounded by the microsporangium. So that microsporangium has four layers in the wall. The first layer is epidermis. The second layer is endothecium. then you have the middle layer, and then you have the tapetum. Right, so these are the 4 layers of the microsporangium. So now the function of the first 3 layer is the protection to the anther plus dehiscence of the anther to release the pollen grains, because the pollen grain has to come out, right? So it helps in the breaking open of the anther to release the pollen grains. So the function of the last layer, tapetum layer, is to nourish the pollen grains. Plus, it produces lipid-rich 𝗣𝗵𝗱𝗀𝗃 bodies. So, these 𝗣𝗵𝗱𝗃𝗃 bodies are required for the secretion of the sporopollenin, so which is the outermost layer of your pollen grain. Right. So, apart from that, the tapetum also releases 𝗘𝗴𝗃𝗃𝗧 enzyme that helps in the dehiscence of the anther. So, these are the functions, and you should also remember tapetum has a dense cytoplasm and it has many nucleus because of endomitosis condition. Right. So, this is about your microsporangium. Now, each of these microsporangium in the center, it has a compactly arranged tissue. So, this compactly arranged tissue is known as your sporogenous tissue. So, this sporogenous tissue has your microspore mother cell. So, this is also known as MMC, or it is also known as the pollen mother cell. So, this pollen mother cell undergoes meiosis to produce your microspores. So, they produce 4 microspores. So, it is known as microspore tetrad. Fine, right? So, this entire process is called as microsporogenesis. Microsporogenesis. The formation of microspores from the microspore mother cell through the process of meiosis is microsporogenesis. So, moving further, so next we'll talk about how the pollen grain further divides. So, each of the microspore, so the microspore is nothing but your pollen grain. So, your microspore is now haploid. So, this undergoes mitotic division to form 2 cells. The 2 cells are vegetative cell and the other one is generative cell. Right. So, generative cell is the bigger of the 2 cells and this one is the generative cell. So, this is the generative cell, this is the vegetative cell. So, in majority of the angiosperms, you can say in 60% of the angiosperms, the cells are released, the pollen grains are released at the 2 cell stage. Fine. In remaining 40% of the angiosperms, the generative cell undergoes further mitotic division, right, and it forms your 2 male gametes. Now, totally how many cells you have? So, you have your vegetative cell plus 2 male gametes. So, this is the 3-cell stage of the pollen. So, this is the 3-celled stage. Fine. So, this is how your pollen grain is released. Now, talking about the structure of the pollen grain. So, the pollen grain is spherical and it's very small in size. So, it has 2 layers. The outer layer is called as XII. The inner layer is called as intine. So, the XII layer is made up of a very resistant material, that is sporopollenin, so which makes the pollen grain look like a living fossil. Now, the next layer is intine. So, intine is made up of cellulose and pectin. Apart from these things, the outer layer XII is also having what is known as germ pores. So, these germ pores are apertures where your sporopollenin is absent and they help in the formation of pollen tube post pollination. Fine. So this is the structure of your pollen grain. So moving forward, so can we preserve the pollen grain? Yes. So you can preserve the pollen grain using a technique known as cryopreservation. So where you preserve the pollen grain in liquid nitrogen at -196 degrees Celsius. So then we also spoke about viability of a pollen grain. So what is a viability? So how long does a pollen grain remain functional after release from the anther? So that is called as viability. So viability varies from one plant to the other. So it is not fixed. For certain plants like your cereals, which includes your rice and maize, so the viability of the pollen grain is just 30 minutes. So within 30 minutes from the release from the anther, it has to land on the stigma, right? So moving further, talking about certain families like your Rosaceae, your Solanaceae, etc. So the viability can go up to 6 months. Thus, the viability varies. So this is what we spoke in the last lecture. So I have given you a recap. Alright, Deepa, fine. So now we go to your Megasporangium. So we come to the female portion, that is pistil. So just like the male portion is called as andrecium, the female portion is called as gynoecium, and just like the male portion is represented by stamen, the female portion is represented by pistil, right? So now pistil has 3 parts. Just like your stamen had 2 parts, your pistil has 3 parts. Hi Rohan, hello, a very happy good evening. So thank you so much for joining my lecture. I hope you're doing good. So please don't forget to like, share and subscribe. So Rohan, did you talk to the customer care center? So did they tell you how to shift from that course to this course? So did you get any updates? Alright, so the pistil has 3 parts, stigma, style and ovary. So I guess you would have seen stigma, style and ovary. So I will show you stigma, style and ovary. So here it is. So this is stigma, style, and this bulged portion is the ovary, right? So stigma is the landing platform for pollen grain. So once pollination happens, your pollen grain lands on the stigma. So stigma is that receptive portion which is ready to take up the pollen grain. So style is an elongated slender which is present just beneath the stigma. Right? So these 2 sentences are clear. Now ovary, so ovary is the basal portion and it is the bulged portion. So that is the definition of ovary. So now moving further, inside the ovary. So the space inside the ovary is known as the ovarian cavity. So this ovarian cavity is also known as locule. The space inside the ovary is known as the ovarian cavity, or it is also known as the locule. Fine? Yes, ma'am, but did not respond. Okay, so did you get in touch with my Telegram group? I can help you out. So please get in touch with my Telegram group. I will ask the customer care to get back to you. Alright, yes, I will also talk to them like what is the procedure, so is it even possible? But get in touch with me in my Telegram group. So I don't know, I haven't checked Telegram, but if you are in touch, yes, I'll get back to you. I will talk to them. Alright, yes. Fine. So the space inside the ovary is the ovarian cavity. It is also called as the locule. So inside the locule, you have a cushion shaped structure that is called as placenta. And to the placenta are attached your ovules. Your ovules are called as megasporangia. So once again, so you have 3 parts of your pistil. So stigma, style and ovary. The place inside the ovary or the space inside the ovary is called as the ovarian cavity or the locule. Okay. So inside the ovarian cavity, you have a cushion shaped structure that is placenta. So this placenta is attached with ovules and the ovules are called as megasporangia. Microsporangia are the pollen sacs. Megasporangia is the ovule. Megasporangium is the ovary. Fine? Right. So the number of ovules can vary. So some plants can have only 1 ovule in case of wheat, paddy, mango, because mango has 1 seed, right? So papaya has many seeds, so they have many ovules. Papaya, watermelon, orchids have many seeds. Okay, Rohan, fine, I'll help you out. It's not an issue. Okay. So now moving further, so megasporangium, so let's talk about it. So the ovule is attached to the placenta by a stalk and that is called as funicle. So here, let me show you. So this is the structure. So here you have so this is your placenta and this is the structure of your ovule. So the ovule is attached to the placenta by a stalk that is called as funicle. So this stalk attaches the placenta. So suppose, assume here I have my placenta and this is my ovule. So it is attached by a stalk that is funicle, right? So the body of the ovule fuses with the funicle and that is hilum. Yes, ma'am, a very happy good evening. So thank you so much for joining our lecture today. So please do like, share and subscribe. Ma'am, Megasporangium are called as pollen mother cell or not? No. Whenever it is mega, it refers to the female portion. So when you say Megasporangium, it is the ovule. So pollen mother cell is present inside the microsporangia. So pollen mother cell is present inside the microsporangia. So it is present in the sporogenous tissue. When you say mega, it comes to the female portion. So here what is the mother cell? Here the mother cell is called as megaspore mother cell. So your megasporangium and pollen mother cell are two different things. So pollen mother cell comes in andrecium, megasporangium comes in your ovule. Okay, so you will not confuse both. Fine. So here for you to make you understand, so you have the placenta. So to the placenta, the ovule is attached. So the ovule is your megasporangium, right? So inside the ovule, you have your megasporangia. So we'll come to that. That will be your embryo sac. Okay. So the placenta and the ovule are attached by a stalk. So this stalk is called as funicle. So the place where your funicle attaches with the ovary, this portion is called as the hilum. Fine. So you understood what is funicle and what is hilum, right? Fine. So each ovule has 1 to 2 protective envelopes. So surrounding the ovule, you have 1 to 2 protective envelope, and that envelope is called as integument. So this can be 1 layer, it can be 2 layer, or it can be 3 layer. So if it is 1 layer, it is known as unitegmic. So if it is 2 layer, so we call it as bitegmic. If it is 3 layer, we call it as tritegmic. But this envelope is called as integument. So if you can see this integument, so you have outer integument and you have inner integument. So this integument surrounds the ovule throughout except at one opening. So except at this opening, it is not covered, right? So this opening is called as the micropyle opening. Fine. So micropyle is exactly opposite to the chalazial end. So this defines the vegetal pole. For example, so when you are born, so for example, you are formed by zygote, right? So initially, before you became a baby, so you were in the form of a single cell zygote. So the zygote has to undergo repeated division and your cell takes up the position, right? So this will become the head and this will become the posterior pole. So this will have your legs. So the poles are decided. So similarly, this pole is the micropyle end, which helps in making the decisions later, and this is your chalazial pole. Alright. So chalazial pole is the basal part of the ovary. Now, inner to your integument, you have a structure called as nucellus. So, inner to your integument, you have nucellus. Fine? So, now we are talking about your megasporangium. So, megasporangium is your ovule. So, megasporangia is the female gametophyte. So, the female gametophyte is the embryo sac. Fine. So, now you have the placenta and you have the ovule. So these two are connected by a stalk and the stalk is called as funicle. So the place where your funicle and your ovule attaches, so that is called as hilum. Fine? Right. So now the ovule is surrounded by 1 or 2 protective envelopes and that protective envelope is called as integument. So integument surrounds the ovule at all the points except at one point. The place where the integument does not surround the ovule. So the place where the integument does not surround the ovule, so that opening is called as the micropyle opening. So exactly opposite to the micropyle opening, you have your chalacal pole. Right? Now, inner to the integument, you have another layer that is called as the nucellus layer. Now, this nucellus layer has what is called as the mother cell. So, this is your megaspore mother cell. So this megaspore mother cell, it undergoes meiosis to form 4 megaspores. Fine. And this entire process is known as megasporogenesis. So in the andrecium, we spoke about microsporogenesis, and this is your megasporogenesis. So nucellus contains your megaspore mother cell. So this undergoes meiotic division to form 4 megaspores. So out of the 4 megaspores, only one of them is functional. The remaining 3 degenerates. So unlike your microspores, where all the 4 microspores are functional, here only 1 megaspore is functional out of the 4, right? So now this 1 megaspore is haploid. So each of the megaspore is haploid. Megaspore mother cell is diploid. Nucellus is diploid. Your integuments are diploid. Fine. So now what is the fate of this megaspore? So this megaspore, which is haploid, undergoes your first mitotic division. So when it undergoes mitotic division, it forms 2-celled stage. It forms 2-cell stage. Right? Now further, so it forms 2 nucleate stage. So now it has 2 nucleus. So further it undergoes the second meiotic division. So that is the 4-celled stage. Further it undergoes third mitotic division. So it forms the 8 nucleated stage. So when this nucleus divides, it forms 2 nucleate stage. So we'll keep it as nucleate stage. So this forms 2 nucleate stage. So this forms 4 nucleate stage, and finally it forms 8 nucleated stage. Now what happens to these 8 nucleated state? So yeah, megasporogenesis. So this is what I've told you, right? So we are here. So this is the micropyle end, the chalacal end. So inside this you have the nucellus. The nucellus has the megaspore mother cell. So this megaspore mother cell first divides into 2. So it forms the megaspore dyad. Right? So then it undergoes further division and it forms megaspore tetrad. So out of this megaspore, only 1 is functional. The other 3 degenerates, right? So it finally forms 8 nucleated stage, right? So out of this 8 nucleated stage, so 6 out of the 8 nucleus forms a cell wall around itself. So now assume this is roughly the structure that we are talking about. So I have 8 nucleus here, right? So 1, 2, 3, 1, 2, 3, 1, 2, 8 nucleus. So out of this, 6 of them forms a cell wall, right? And out of the 6, 3 cells, they move towards the chalacal end, and 3 cells move towards the micropyle end. Fine? and the two central cells are here. So this is known as secondary polar nuclei. So the three cells at the 𝗘𝗴𝗃 end, so this is the 𝗘𝗴𝗃 end. So the three cells at the 𝗘𝗴𝗃 end are called as antipodal cells. Fine. The three cells towards the micropyle end, they are known as your egg apparatus. So in the egg apparatus, I have one egg cell. So one is my egg cell and the other two are known as synergids. So this entire structure of 8 nucleated, so this is totally how many nucleate? So this is 8 nucleate, 1, 2, 3, 4, 5, 6, 7, 8. So it is 8 nucleated, but how many cells are there? 1, 2, 3, 4, 5, 6 and this entire one. So 8 nucleated, 7 celled embryo sac. This is your female gametophyte. So this is the structure of the female portion. So we spoke about the structure of the pollen grain and this is the structure of the egg apparatus or the embryo sac in detail. So the female gametophyte, only one is functional. So you have the one functional megaspore nucleus. It undergoes mitotic division to form 2 nuclei. Further, it undergoes mitotic division to form 2 nucleate embryo sac. Then further mitotic division to form 4. Then further it undergoes mitotic division to form 8 nucleate. So immediately it does not form a cell wall. So it takes time. Right? So initially it forms 2. Then undergoes further division, it forms 4. So further division, it forms 8. So 3 move to one pole and 3 moves to the other pole. So 2 remains in the center. So this 3 towards the 𝗘𝗴𝗃 end, it forms the cell wall. So that is called as the antipodal. And towards the micropyle end, it forms 3 cells. So that is your egg apparatus. In that you have 1 egg cell and 2 synergids. And towards the synergid, you can see finger-like structures. So this is called as filiform apparatus. So remember what is a filiform apparatus? I will explain when we are talking about fertilization, right? And these two central things are called as the polar nuclei. So together this entire thing is 8 nucleated and 7 cell. So the embryo sac is 8 nucleate and 7 cell. So the embryo sac is known as the female gametophyte. So today we spoke about the female portion. So we are done talking about the male and female gamete. So the next class we are going to talk about how pollination happens. So next class, that is day after tomorrow on Friday, we'll talk about what is pollination, what are the types of pollination, what are the agents of pollination and what are the advantages of pollination, how to prevent self-pollination, how to promote cross-pollination. So the entire concept of pollination will be covered in one lecture. So please do join, because in pollination you have a lot of questions. So for term 1 as well as for your NEET exam. Right? So please join me for the same. So I hope you guys understood what is Megasporangium. So this is my last slide. So we are done with today's topic. So if you genuinely liked my way of teaching, so please do like, share and subscribe. So for all those who are new and wondering why ma'am is saying day after tomorrow, so I teach class 11 and class 12 on an alternate basis. So Monday, Wednesday, Friday I teach class 12, Tuesday, Thursday, Saturday I teach class 11. So in class 11 I am dealing about transport in plants. So yesterday we spoke about transport in plants, which we will continue tomorrow at 5:15 p.m. So day after tomorrow I will talk about pollination. Yes, Natasha, so thank you so much for joining my lecture. So I would request you all to come for tomorrow's lecture at 5:15 as well. So until then, stay tuned and take care. Bye.